this chance to talk about CRISPR-Cas tools and how it is assisting plant disease management. So I'm not going to detail introduction of this one, but this is the outline of my today's talk. In part one, I will be talking about a little bit of methods, what is genome editing and why we need that, and how CRISPR-Cas9 edits the genome, brief mechanism. And in part two, I will be giving you some case studies where CRISPR-Cas9 has been used for plant disease management, even improving host resistance. And in part three, I will be talking about briefly in 2 or 3 slides about disease diagnosis. So these tools are also being used as a diagnostic tool. And in part 4, I will be going to advanced genome editing technology that is known as base editing. Okay, so this is a higher eukaryote like human being, and this is an example of rice plant that is also higher eukaryote, and they are made up of trillion of cells. And if I want to give you an example of what are the cells, this is a plant cell, example of plant cell, and that has a nucleus. And this nucleus contains, this is the head of the cell because it contains chromosome. And this chromosome is nothing but a coil of a long thread of DNA. But according to the present status of knowledge, the whole thread of DNA is not equally important. So a functional unit or a functional portion of that long DNA is called a gene. And why this gene is a functional unit? Because when a gene expresses, it produces some protein, and that protein governs a character or a phenotype of a plant or an animal or any organism. So a gene is a functional unit of a DNA that produces a character. Now, what is in the gene or what is in the DNA? The DNA has nothing but 4 different letters, A, G, C, and T, and the sequence of those letters actually makes up the genetic code of an organism, and that controls almost everything in an organism. So if I want to introduce genome editing to you, this is a very good slide to understand. So this is a classical example of host pathogen interaction between rice and bacterial blight pathogen, that is Xanthomonas oryzae. So this green box is the rice cell, and this violet one is a gene that is called sweet gene. This gene produces a transporter protein. This transporter protein exports sucrose from inside the cell to the apoplastic region, to the outside of the cell. Okay, so this is the normal function of this gene. But now, this bacteria is very, very clever, and they uses this gene for their own benefit. How? This Jew bacteria, when they infect rice plant at the initial point, they secrete a protein that is called TAL effector. This TAL protein or TAL effector binds to the promoter of that gene. Any gene is controlled by a sequence that is called promoter that actually regulates its expression. So when this protein secreted from the bacteria binds with the promoter of that gene, then the gene expression goes higher. And when the gene expresses higher, as I told you, it produces protein. So the more protein will be produced, and there will be more transporter in the membrane. Now, as a result of this more number of transporter, the sucrose will be transported more amount outside of the cell. So now the bacteria is very happy because this bacteria uses sucrose or other sugars as their nutrition. So as they have abundance of nutrition, they will grow vigorously and they will infect more that rice plant. So this is how they uses rice gene for their own benefit. So now the question is, you can easily hypothesize with me that if I want to show you a genome view of that region, this is sweet gene and this is the promoter, but this effector protein secreted from the bacteria does not bind anywhere in the promoter, but it binds to a specific position of the promoter, and that position is called effector binding elements. This is a sequence in the promoter. Okay, so now you could hypothesize with me if we could hamper this interaction between effector and these element of the promoter, so we could probably develop bacterial blight resistance. Okay, so the question here is, can we specifically disrupt the EBE sequence in the promoter? The answer is yes. By genome editing, you could do specifically disrupt that kind of sequence, but not hampering other portion of the genome. So suppose this region is the EV sequence of the sweet promoter. Now you want to lock that so that it won't bind to the effector protein secreted from the bacteria. Okay, so that's the way that is called genome editing. Now, if I want to give you the most popular genome editing tools, that is CRISPR-Cas9, there are other tools that has been developed earlier, but I'm not going into detail. So this CRISPR-Cas9 can cause targeted inactivation or targeted changes in the genome sequence, in the sequence of any region of your genome. So if I want to give you a very easy example to understand how this technology works, it has two different components. One is a customizable RNA that is called guide RNA. So you can design your own RNA. And another is a DNA cutting protein that is called Cas9. So this protein can cut DNA. Now, if I want to give you a very easy analogy, so this guide RNA is nothing but like a genomic address. So you are giving an address and you are writing that address because you can customize this RNA. And now this Cas9 is like a postman, okay? So this postman has two scissors in its pocket. Now, when you give the address to the postman protein takes the address and it goes to the genome and it finds similar address. If it finds similar sequences as in the RNA that you gave, so then it will search for the similar sequence and then bind to that similar sequence. And whenever there is an exact binding of this postman with the genomic sequence, then it gets the signal. So this is the position where I have to make a cut, because this postman has two scissors in its pocket. Now, one scissor cuts one strand of DNA. As we know, DNA is a double-stranded structure. So one scissor cuts one strand, another scissor cuts another strand. So it makes a double-strand break in that targeted DNA. Now, it's clear that you could use CRISPR-Cas9 to make a double-strand break at a defined position that you want to, okay? Now, how it actually creates mutation? The answer is here. So whenever there is a DNA double-strand break, this kind of break cell treats this kind of break very serious threat to them because it's a genetic material, okay? So cell immediately tries to repair that double-strand break. And there are two pathways. One is NHEJ, which is the predominant pathway. Cell, in most cases, apply this pathway to religate the broken end, okay? Now, this NHEJ is also error-prone. It can introduce errors in the genome. So inadvertently, it can introduce some new nucleotide or new bases at the junction, or also can delete some nucleotides from the junction. So this rate is actually showed here as a random insertion or deletion during the religation procedure or during the repair. And due to this random new insertion or an original deletion, then the open reading frame of that gene becomes disrupted. The sequence is disrupted. As I told you, sequence is everything, okay? So sequence of that 4 letters AGCT for that gene is disrupted now. So the gene is now non-functional. This gene cannot function in the same way as it was doing during its original form, okay? Now, from this video, you would be understanding how it actually works. Okay, So now coming to the second part of my presentation, I will be giving you briefly a few case studies because time is short. So, you know, as I told you already, this example of sweet gene. So sweet gene is present in rice plant, but it helps bacteria to infect. So this kind of gene is called susceptibility gene. So because this gene helps in early pathogen establishment, and it can also negatively modulate the host defense. So now, as I told you, if we could disrupt EBE with genome editing, then the TAL effector would not be able to bind to the EBE sequence. So the plant would be resistant to that bacteria. Okay, so there are a lot of reports, but I highlighted these 4 reports, recent reports, and they have showed a very good level of resistance with editing of this EBE sequence. So you can see here, this left panel is the control one that is not edited, but these 3 are with edited EBE sequences with CRISPR-Cas9 and TALEN, and you can see level of resistance. This is full leaf length is infected with the bacterial-like pathogen, but this is very less amount of infection. So this is a very classical example of that kind of things. If I talk to fungal infection, also in case of wheat and tomato, they are also, sorry. Oh, okay. So is it visible, Bennett?
Slides are visible.
Yeah.
Yes, sir.
Okay, thank you. So for wheat and tomato, also there are susceptibility genes that's called powdery mildew locus, MLO. This MLO gene also helps the pathogen to infect powdery mildew pathogen. For wheat, there are it has been genome edited, this locus MLO gene, and also for tomato, and they have shown to impart a great resistance. You can see this is the wild type infected hugely with the powdery mildew fungus, but here the one which is edited, which is not infected. So coming to so coming to viral example of viral disease resistance using this technology. So you could develop an immune system against virus. How? Because in the plant cell, if you express Cas9, the postman that I told you, and its address, that is guide RNA, and this address would be to target the viral DNA that is most likely to infect that plant. Okay, so if you express this Cas9 and guide RNA in the plant cell, and then whenever it would remain vigilant inside the cell, and whenever there would be an infection from the virus, you know, virus is known to inject its genetic material, either DNA or RNA, inside the cell. Okay, so whenever it injects this DNA inside the cell, then this Cas9 and guide RNA complex, they go to that cell because you have given that address. Okay, go to the viral DNA and it will bind there and it will chop the viral DNA so that the virus would not be able to replicate more. So it would give you to create an artificial immune system against a particular virus. Okay, the next question you would be asking me, okay, virus genetic material, maybe DNA or RNA, you have given me example of DNA, but what if the virus contains an RNA virus? So there is also in the toolbox, because this toolbox, you know, CRISPR-Cas toolbox is expanding in a very, very faster way. And this Cas13 is a protein, which is a brother protein of that Cas9 that I talked to. And this Cas13 has a similar role, but only distinction is that this Cas13 can go and bind to RNA and chop RNA. So instead of using Cas9, you have to use Cas13, and you keep the address, same guide RNA address, and then you can use it against any viral RNA virus. Okay. Now coming to the third part of my presentation, this is disease diagnosis. So, you know, disease diagnosis is also an emerging area where CRISPR is being used. So this is, you know, Jennifer Doudna, who got Nobel Prize this year. So, you know, this Cas12 is another kind of protein. That's orthologous Cas protein, and this protein has a unique properties. It obviously goes and binds to the target and cut the target, but it has collateral activity. Collateral activity means when it finds its target, then it will bind and chop that target. But it also can, after it finds its target, it also can chop other single-stranded DNA if it is present in the vicinity of that protein. Okay, so these actually properties of this protein has been used in detection method. So what they have done, suppose you have a tube and that tube has some viral DNA, and you want to detect whether the Cas12 can detect that virus DNA in the tube or not. So you add Cas12a, and then you add address like guide RNA, and also you have to add this kind of single-stranded DNA, which is linked with a quencher dye and a reporter dye. So what happens when there is a quencher and reporter combination, then this reporter dye would not fluoresce, okay? But if they are separated, then it would fluoresce. So now, in the tube, you applied Cas12a, you applied guide RNA, and you have a sample with the viral DNA. Now, whenever it finds a DNA target, it will cut the DNA target. And as I told you, it has collateral activity. So it will also cut the quencher and reporter single-stranded DNA, and so that the quencher would be separated from the reporter, and then the reporter would fluoresce. And this fluorescence would be detected as an indication of presence of your nucleic acid of interest, like viral nucleic acid, any bacterial or fungal. So this method is called detector. And another similar kind of thing for RNA detection, this protein could be used, that is Cas13. And that method is called Sharda method, that has been developed by Feng Zhang from MIT. And it has been already approved by FDA in the US and has been used in California for detecting coronavirus, because coronavirus is RNA virus. So similarly, it has been also being used in plant for trait detection, pest surveillance, and pathogen identification. So this is an emerging area in detection technology. So this is the last part of my presentation, and I'll quickly go through this part, and this is the most exciting part, and that's why I kept it at the last of my presentation. So now single base editing. The question comes, so this is in a snapshot I have taken from the famous movie Power, which has been led by Amitabh Bachchan, and this is Oro. Oro is only 12 years old, but he looks like 70 years old, even more. So because he is suffering from a rare genetic disease, and this disease is called progeria. Progeria causes rapid aging at childhood, and they look like an aged person. And you can see they are both equal aged. His friend also equal aged, but what is the difference between these two? Difference is that this Oro's friend has G at a specific position, and this G is not present in case of Oro, but A is present. Because of the single changes in the gene sequence, Oro has died at 13, and this is the case of many progeria infected children all over the world. So we knew the reason behind the suffering, but we did not have any cure. So if I want to give you a plant perspective, this is the same genotype. This is Kitachia. It's susceptible to a fungal disease because of having a C nucleotide. But if it has a T nucleotide, it becomes a mutant, and it would be resistant to pathogen. So these single nucleotide differences also determine susceptibility and resistance. So the question here, is there a way to rewrite specifically genetically? Is not disrupting a gene, as I showed you with the conventional CRISPR-Cas9 technology, but you have to rewrite C to T or A to G, okay? So the answer is yes, because you could do point mutation with base editing tool. This is called base editing, and this is a CRISPR-derived tools and has been invented by David Liu from MIT. And there are two types of base editor widely used. One is adenine base editor that can convert A to G. Another is cytosine base editor that can convert C to T. Now, this video will make you clear. Okay, so what we tried when this technology came, so we thought to apply it for plants. So we tried to apply adding in base editor in rice, and we made several factors. And what we did, we tried to target two different single nucleotide mutation at two different genes in a single go. So the first gene was WSL5 and another was Z3. And I'm not going into details of this complex technology. So what we did, we fused both the address in a single RNA and fused with tRNA also for increasing the transcription rate. So what it would produce like this: WSL5 guide RNA or the address would be separated, and it would take the base editor and take to the chromosome 4, because WSL5 is present in chromosome 4. So the editing should be done in chromosome 4. Similarly, Z3 would be separated from the address and it would go to the chromosome 3 because Z3 gene is present in chromosome 3. So this is called multiplex base editing. So now coming to the exact final result of this, I'm not going into details of those things. So this is the control one, this is the wild type one. So our target was this A nucleotide in the first exon of WSL5 gene. You can see this is a Sanger chromatogram. This is very easy to understand. You know, blue means C nucleotide, green is A, and black is G, and red is T. So this green pig, A nucleotide, we tried to change it to a G nucleotide. So when we treated our rice plant with the base editor, then we have found very exciting result that this A has been converted specifically and very precisely to a G nucleotide. And you can see the other pigs has not been changed, only the target A has been changed. So this single nucleotide change we could artificially create with the base editor, and we got the similar mutant phenotype as expected in the as reported earlier in the literature. So this single nucleotide mutation is reported to cause this white stripe leaf phenotype. So this is a white stripe leaf in the mutant that has been created with this base editing. So now coming to the second gene, the similar example, this A, I targeted this A, we targeted this A and tried to convert it to G, and you can see this is the mutant one, and we could convert precisely this A nucleotide to a G. And similarly, other nucleotide in the adjacent region of the genome has not been changed. And similarly, we got the expected result here. Z3 is nothing but a zebra 3 mutant. There's a light green, dark green strap in the leaf, and also it shows dwarf phenotype. So those all expected phenotype we got because of the single mutation that we could make with the base editing. So if you know any SNP that causes a very good character, you could alter those SNP with this base editing technology. So a few slides left. So the main question is why people are talking about genome editing, because and they call it non-GMO, because genome editing reagent, when it enters into the cell and it causes its editing, then the reagent is no longer needed. Okay, so if I want to give you an example here, suppose base editor you incorporated into rice plant, and it is suppose it is integrated into chromosome 1 and your target is in chromosome 4. Now this base editor construct that you transform, it will produce the protein here and it will go to the target, okay, and it will make the mutation. So once the target is mutated, you can easily understand that this base editor is no longer needed. So by virtue of sexual segregation, if you grow next generation, then this base editor would be separated out and you select only for the mutant, not for the base editor. So you have to select transgene free mutant. So if I want to see one practical example, this is 25 mutant plants, and you can see the 6 plants did not have any base editor in the next generation. So those plants are transgene free, but they are mutated. So that's how we can obtain very quickly transgene free and mutated plants. So this is another exciting single slide. So this is a classical example of how susceptibility could be converted to resistance. So in one gene there is a premature stop codon. So in cellular DNA sequence, if TAG is present, that is treated as a red signal to the translating machinery. And this TAG is a stop codon, so protein synthesis would be stopped here. And as a result, if this TAG is present, the protein is not produced in full length, and the genotype would be disease susceptible. Now what we did, we applied base editor to convert TAG to a TGG, and when we applied, we have seen, you can see from this result that we could obtain a conversion of TAG to TGG. So TGG is not a stop codon now. It's now a green signal for producing this machinery. So now it would produce the whole length of protein and the plant would be disease resistant. So it has not been published, but the work is going on. So in order to know more, so you could go through in details of this base editing technology by our recent review in Trends in Biotech. This is another one. And that I have not talked about. I talked about adenine to guanine and cytosine to thymine base editor, but recently there are 3 reports, very quick succession, and we have wrote a highlight article for those base editors that could convert a C nucleotide to a G nucleotide, cytosine to guanine. So you could also go through this one. And this is a recent edited book published, and where we jotted down all the protocols for applying this excellent technology to different crops, even in bacterial, fungus, and human mammalian cells also. So you can go through those things. Thank you very much. I know I tried to be precise. Thank you.
Yes, sir. Thank you, Dr. Kutubuddin, for your wonderful presentation and for sharing your research work with us on specifically on Cas9 technique and how genome editing is contribution in this field of agriculture. And I think we have a lot of students who are here in our participants, and they will be probably more encouraged to work in these aspects which you have shared with us. I would just quickly like to ask the audiences if they have any questions to ask to Dr. Kutubuddin. Okay, sir, I have one query regarding this Cas9 gene technique, that as we said, we are looking it on from a disease point of view, like how it is interacting. So is there any other functionalities of this Cas9 technique, like we have when we talk about a crop? So there are different functionalities from other point of views from besides disease. So is Cas9 also looking into that point of view?
Exactly. So this is a huge technology, and that's why, Nobel committee takes only how many years? 8 years to give Nobel to Jennifer Doudna and Emmanuel Charpentier, because it has huge implications, including agriculture, medicine, and human therapeutics, and basic biology also. So I just provided, as my title goes with the title, those examples I have given, but this is not herbicide tolerance is being generated with CRISPR-Cas9. And then suppose very simple example, that suppose rice has phytic acid as an anti-nutrient factor. So you want to develop a low phytate rice. So you know that phytic acid biosynthetic pathway, and if you could modulate, apply CRISPR-Cas9 of that biosynthetic pathway, you could produce a low phytate rice. Similarly, low phytate maize. So this is another example. So plant disease is a sector of application, but there are a lot more. So suppose negative regulator of yield also. There are a lot of genes known to regulate negatively towards yield of any plant. So those negative regulators could also be disrupted with CRISPR-Cas9. So this is for disrupting. But if you want to change a SNP, single nucleotide mutation, that is a functional changes, so you could also do that with base editing. Yeah.
Thank you, sir. I think the book which you have shared.
Thank you, Dr.
Yes,
Kutubuddin. It was a wonderful talk, really. A lot of new technology that you explained. And I'm sure this will also be applicable to induction of resistance or tolerance in plants to various biotic stress factors, or maybe pathogens.
Yes.
Maybe, in fact, one day something, I think we'll arrange for you something specific talk on this aspect, resistance mechanisms of plants or how to modulate them.
Yes,
and perhaps that would be a good idea. Someday later, maybe individual talk by you.
Yes, sir.
That would be great. Thank you.
Thank you, sir.
Thank you, sir. Now I would like to invite